529 DNA polymerase, which has 5 prime to 3 prime polymerization activity and 3 prime to 5 prime single stranded DNA exonucleotide activity. So this is how I think all of you know about the RCA. So when we have a single primer, this is how the RCA amplification takes place. Since we are going to use 529 DNA polymerase, it has again a stand displacement ability. So we get a long stretch of DNA formed. So instead of one, we can use multiple primers. So then we get a more amount of nucleic acid formed. Then if you use a unique restriction enzyme for this particular virus, so we can get the individual full length genome of this virus. So the major steps includes the isolation of DNA from the plant, denaturing it, and setting up the RCA using either random primers or specific primers, or combination of both random and specific primers. Then incubating for about 14 to 16 hours at 37, and then you subject the RCA product for restriction using unique restriction site for the target virus and analyze on the agarose gel. So presence of expected size product indicates a positive reaction, which can be confirmed by cloning and sequencing. So the another advantage of this RCA is that it can be used to identify or diagnose novel DNA viruses, circular DNA viruses. So the third method, isothermal method, is the recombinase polymerase amplification. So this is very rapid isothermal amplification methods. Amplification efficiency is quite high and requires just about 15 to 20 minutes. It uses a combination of enzymes and proteins such as recombinase, single-stranded binding proteins, and stand displacing polymerase. So employs a pair of primers which combine with the recombinase that targets the double-strand DNA and facilitates stand displacement. So just we can see here. So this is the your template. So this primers and the recombinase protein complex is added. They form a complex. This complex takes it to the homologous region in the DNA, and then the recombinase destabilizes the stand here. So the primer will bind. When the primer binds, the single-stranded DNA binding proteins will stabilize this region, and then the stand displacement polymerase extends the chain. So that's how this amplification takes place. So it is like a lamp. This is also done at 37 to 42 degrees centigrade. It requires as little as 10 to 15 minutes. It's highly sensitive. Yeah, performing army is you have a usually this is, comes with a twist. Is the one company which is manufacturing the components required for this. Now coming to the my application of this, all the methods for the detection of viruses in the spices. So as we all know, Western Ghats of India is the center of origin for important spices such as black pepper and cardamom, and viruses are important production constants in both these crops. And coming to the black pepper, we have an important viral disease called stunted disease in the black pepper. So this is the kind of symptoms we see. All kinds of symptoms like mosaic, brittling of leaves, smalling, twisting and rolling of leaves. As the name indicates, finally the whole vine will get stunted. So there are two viruses involved in this. One a single-stranded RNA virus, a cucumber mosaic virus. So we have completely sequenced this virus. It's about 8.6 kb in size. And then we have a Badna virus associated with this disease. It's a double-stranded DNA virus, Piper yellow mottle virus. It is transmitted by mealybug. And this is the complete genome of this virus. It has 4 ORF with then separated by intergenic region. Coming to the viral disease of cardamom, we have 3 important RNA viruses infecting cardamom. One is the mosaic. It's also called as cut tear marble, caused by a macular virus called cardamom mosaic virus. Then we have recently found out another virus called chlorotic streak, caused by a strain of banana bract mosaic virus in 2012, which is again a potty virus. Then recently we have characterized the vein clearing or Kokekandu disease of cardamom, caused by cardamom vein clearing virus, which is a new species in the genus Nucleorhabdovirus. So this is about the cardamom mosaic virus. Madurai Kamaraj University persons have completely sequenced the genome of this virus. It is 8249 nucleotides long. This is the, you find lots of variability in the symptoms, both variety wise as well as region wise. This is the chlorotic streak. So unlike Katte, so this causes intravenal chlorosis. So very easily can be distinguished from the mosaic disease. So we have completely sequenced this virus as well. And then we have the third virus, the Kokkekando, as I said, it is the, it's in endemic in certain districts of Karnataka where cardamom is grown. It's transmitted by aphid. We have also completely sequenced this virus. It's a, as I said, it's a negative sense RNA belonging to the nucleorhabdovirus. So typical genome organization of nucleorhabdovirus followed a nucleocapsid, phosphoprotein, P3, mastic protein, glycoprotein, and the polymerase. Now, coming to the viruses, the diagnostic, as I said, both black pepper and cardamom are vegetatively propagated. So diagnostics is important. So we have developed both LAMP-based as well as the RPA-based assays for the detection of viruses infecting both black pepper and cardamom. So we can see here the optimization of the LAMP for the detection of pepper viruses. So here we see this is the positive control, this is the negative control, this is the healthy control. So where we could see the positive reaction, the turbidity, the calcium added fluorescence, and then pellet and the gel. In the gel, we can see the adult structure only in the case of positive virus infected plants. So this is the optimization. As I said, we have optimized the temperature here. So we can see here the lamp can be performed at 59, 62, or 65. So in all cases, we got the same results. So this is the time. We have optimized the time. As we see here, 45 minutes with very little amplification, but we get good amplification at 60. And by keeping up to 75, we are not going to get any additional benefit. So within 1 hour, the entire lamp can be completed. So this is the optimization of the magnesium sulfate and the betaine. You can see here, again, these are the optimization of the these two parameters, as I said earlier. And then we also compared the lamp with the conventional PCR as well as the real-time PCR. What we found is that actually real-time PCR is most sensitive, followed by the lamp, and then followed by the conventional PCR. So lamp is about 10 to 100 times more sensitive than the conventional PCR, and it's about 100 times less sensitive compared to the real-time PCR. So this is again the standardization for the detection of BBRMV and cardamom. So again, this is the optimized component for the lamp. So again, the comparison of the sensitivity. Here also we found the same order, that is real time is most sensitive, followed by LAMP, and then least sensitive is the RT-PCR. So this is the standardization of RPA for the for the detection of Piper yellow mottle virus in black pepper. So we'll see the optimization of the, we selected the primer. This is the optimization of magnesium acetate. This is the time optimization and at the temperature optimization. So this is the for the cardamom vein clearing virus development of both RT lamp assay optimization. You will see this is the optimization of the RT RPA, and you will see here the comparison of sensitivity of detection of RT RPA, RT PCR, RT lamp, and RT RPA. So here also we will see that we found real time is the most sensitive, followed by RT lamp, RT RPA, and then the conventional PCR. So this is the, again, we went for validation of the assay by taking field samples. So these are some of the publications we have. This is the published in journal Virological Methods on the pepper, both CMV as well as the Piper yellow mottle virus. This is about the banana bract mosaic in cardamom lamp reaction. That's the about the lamp and RPA for the vein clearing virus, and that's the RPA for Piper yellow mottle virus, and this is again RT-RPA and the LAMP for the ginger virus. So to conclude, I would like to say that viruses are important pathogens of plants. So in view of systemic nature of viruses and lack of chemicals to control, development of sensitive diagnostic assays are needed to check spread of viruses. So isothermal amplification based assays are simple, easy, and provide results quickly. So these methods are suited to resource poor laboratories. Thank you. I think I finished within time. Chairperson.
Just 30 seconds exceeded.
Oh, because yeah.
No problem, because I was telling by the time you come to summary slide, so I kept quiet.
Thank you.
So now it's open for 2 to 3 questions.
என்ன பண்ணும் ஓப்பன் தானே இருக்கு.
Any clarifications or questions from audience? No.
Sir,
Yes.
me,
Shankar.
sir, say it is we have to depend on kit for RPA. Then any kits for available, publicly available kits? Because we have to depend on only one company RPA kit.
Yeah, as of now, it is the only one company is available because we know that it's a patented technology and they have license to fix. Actually, that is the only company as of now manufacturing this product.
Sir, myself Radhika.
Yeah, Radhika.
Sir, can you just give me the cost with respect to all these techniques? How much will be the cost involved?
Yeah, see, the lamp is as good as a PCR only. There is no extra cost involved except for the primers. As I said, minimum 4 primers are required. So PCR also requires 2 primers. So one extra primer is required for lamp, but it is substituted by, as I said, does not require any equipments. Okay, so that's the advantage here. And running time is very less, more sensitive. Okay, and then we can visualize the results by adding some colors or by naked eye. So cost can be increased by if you go by adding colors, or if you want to make it through lateral flow also it can be divided. So it will add to the cost. But a basic or a minimum lamp, I would say it is as good as a PCR. So the cost is quite reasonable. And advantage, as I said, no equipments are required. It can be done even in a, as I said, an agronomy lab or a soil science lab or anywhere, just because. And another added advantage of lamp is that it does not require a very highly purified nucleic acid like PCR. Okay, the crude DNA, they say protocols are there where just 2 minutes or 5 minutes DNA, crude DNA, where people have used that as a template and they were successful in getting it. We have done for RPA as well as LAMP, the crude DNA, just about 5 minutes of preparation, which works really good for detection. That's how we'll be definitely saving cost on this, time on this, and then sensitivity is more. That's what.
Dr. Thangavelu, please mute it.
And sir, with respect to RNA viruses, RNA means it denatures very fast, no? So the same thing goes on for RNA also. Like DNA crude extraction will do for RNA also.
No, RNA you have to isolate the RNA, because crude RNA there are protocols, but we have never standards for this. There are protocols available where even the crude RNA they have used for the detection of viruses using this RPA, especially RPA. RT lamp, I have not seen many articles, but RPA, yes, it is there.
Then we have to go for the normal procedure with trizol or the kit and go for good quality RNA.
Yeah, correct. But I said I can share you some of the articles where they used crude RNA also in the case of what RPA.
Thank you, sir. Thank you.
Ishwar, there is one question in chat box.
Okay.
Do you provide diagnostic service to farmers for seed material certification? This is by Jairaj, who has given keynote address.
Yeah, yes, we were in the DBT's NCS TCP earlier, sir. So we used to provide this service. Now we are not in the NCS TPs, but however, it is we can definitely test the samples and provide the results. It is now on the charge basis.
Ishwar, this RPA, that how long this will be under patent category?
Yeah, because it is 2011, 12, I think it's because it will be still under the patent category now. Lamp is now gone because it it was 2000 they have patented it. So it's
15 years
time is over. It will be over this year. Yeah, it's over. I would say it's over. Whereas RPA will still maybe another 10 years it may be there.
But sir, what about the CRISPR based lamp?
No. We have not yet optimized the CRISPR. Of course, yes, that will be the future. We have to see. Articles say that they are quite cheap and more sensitive. So because we have seen more even for coronavirus detection also, now people have come up with the CRISPR based diagnostic tool. We have not optimized it. Yes, definitely we have to look into that in the future. If it is going to be cheaper, definitely that will be quite good.
Thank you, sir.
So, Chairman, sir.
Yeah, if no more questions, we invite Dr. Thangavel.
Thank you, Ishwar, for your nice presentation.
Thank you.
Okay, now.
Thank you.
So now I request all the participants to keep it in mute mode so that the speaker, when speaking, we can hear it very properly. Now I request Dr. Thangavelu to present his talk on current status on distribution, diagnosis, and management of Fusarium wilt disease in banana. As you are all aware that Thangavelu is a well-known pathologist and well-known banana pathologist working across the globe. He is famous for this TR4 research findings, and I hope he will be sharing that aspect also along with his presentation. Dr. Thangavelu, please.
Yeah, thank you, Dr. Mahesh Kumar, for your nice introduction. Good afternoon to all of you. So now I will present my presentation on current status on distribution, diagnosis, and management of Fusarium wilt, a devastating disease in banana. India is bestowed with blessed with a lot of bananas. We have collected more than 360 accessions belong to different genomic groups. Out of that, more than 20 commercial varieties we grow in India. And we have got a lot of ornamental bananas also, which we are concentrating now as flowers. And in India, except 5 states, we grow banana in all the states except 5 states. And even in Punjab also, the people started growing. Uttar Pradesh, it is grown in more than 1 lakh hectares. So it is growing day by day. The banana growing in different states are growing day by day. And out of different varieties what we grow, this Cavendish, it occupies 63% of the area for banana production. And this we are exporting to foreign countries. And similarly, the Nendran, we are exporting. Then red banana also now we started exporting. So this since this 63% Cavendish, this is being attacked by Fusarium wilt, we are more bothered. Otherwise, this Fusarium wilt was there since 1914. So in different varieties like silk group and other Mandan group. But since Cavendish is now attacked by the tropical race 4, we are bothering more. So let us concentrate on this. And we started exporting banana also. Recently, NRCB has developed a technology and started exporting to distant markets like Europe and Gulf countries. And I am also one of the team members in that. This Fusarium wilt, everybody may be knowing about this because it is a soil-borne disease. Once it enters into the field, it is very difficult to eradicate. If it is, we have to manage. Like COVID, we have to live with the COVID. So we have to live with the Fusarium wilt of banana. So we have to be more careful not to allow the Fusarium wilt to enter into the field, since it survives as a chlamydospores for more than 4 to 5 decades in the soil. So this is with regard to the incidence of Fusarium wilt disease in Cavendish group. This is Cavendish group alone. In different states, you can see here in Bihar, Uttar Pradesh, West Bengal, then Madhya Pradesh, Gujarat, Maharashtra, and Tamil Nadu. We have recorded the wilt disease in Cavendish. And if you just take 20% as a wilt incidence, we incur about 5,000 crores per year loss. Every year we lose 5,000 crores of rupees because of Fusarium wilt disease. This shows the importance of disease which we have to control it. And if you see, everybody, most of the pathologists may know about the presence of these races, different races in FOC, race 1, 2, 3, and 4. The race 1 should not, supposed not to attack Cavendish. Race 4, this is attacking Cavendish throughout the world, and more than 20 countries they have reported the tropical race 4, particularly tropical race 4, and the race 4 group attacking Cavendish. But unusually, we have recorded the incidence of visceral disease in Cavendish due to race 1. 3 different VCGs we have recorded: 0124, 0125, and 01220. This I will tell you right now. This is, you can see here, the Cavendish plantations devastated due to race 1 in 10 districts of Tamil Nadu. You can see the symptoms, and here in 10 districts, the incidence you can see here 5 to 25% incidence is there. Now, even 100% we have noted maximum incidence in 10 districts due to fission filter. And this is about the confirmation due to by VCG volatile production. And this is about the first report of this tropical race 4 in India. This we have recorded in 2015 itself, but we could publish only in 2019 because of the restriction by ICIR. And this is the incidence in Bihar, and the pathogenicity and the VCG analysis. This is the 0121316. This is the specific VCG belong to tropical race 4. And this is about the PCR technique for the confirmation. And we have done the whole genome sequencing, and this shows the difference between the race 1 attacking the Cavendish and the tropical race 4 attacking the Cavendish. You can see the genome size difference, and also in the secretome genes, you can see the difference in FITAR genes as well. So there are differences between the race 1 and tropical race 4 infecting Cavendish. And this is about the comparison between the FOC race 1 and TR4. And you can see the gap in chromosome 3, 6, 14, and 15. And in the FOC race 1 TR4 with reference to the reference FOC taken in NCBI. So these are all the unique genes present in FOC race 1. FITAR genes, unique FITAR genes present in FOC race 1. And also we have identified the unique genes, FITAR genes present in the FOC TR4. There are about more than 30 to 40 genes. And this is, you can see here the difference in FOC race 1. In the case of SAX genes, secreted in xylem genes, this is the FITAR genes, which is very important for the pathogenicity. This is our sample, and this is the reference genes. So 1 is there, but this 4 and 13 in all VCGs of FOC, this should be there. But in the case of our FOC race 1, which is attacking Cavendish, it is not there. Likewise, this 8A, B, it should not be there. This should be present only in tropical race 4, and this is not there. So there are some differences in SIX 4 and 13 in our FOC race 1. And similarly in TR4, tropical race 4, very peculiarly, this you can see here, A should be there in tropical race 4, and B should be in there in subtropical race 4. But in the case of actually this in our tropical race 4, both the genes, FITAR genes of A and B are there. The B should not be there in tropical race 4, but since TR4 is there in subtropical regions of India, that is Bihar and Uttar Pradesh, maybe this SAX B also there. This is very specific to subtropical race 4, but this is present in tropical race 4 of our Indian isolates. So this is the major difference what we have noted. And recently we have recorded, apart from 0124 attacking the Cavendish group belong to race 1 in 10 districts, we recently we have collected the FOC samples from central part of India, central part and northern part of India, and we have collected FOC samples from Cavendish alone, Bihar, Uttar Pradesh, Gujarat, and also in Tamil Nadu. And we have characterized, and the FOC isolates collected from Cavendish caused the symptoms in tissue culture plants of Cavendish, and you can see the symptoms also, both internal and external symptoms. And when we have done the VCG analysis, surprisingly, we have noted the presence of 0125 and 0120 of which belong to race 1. This actually everybody we feel that the tropical race 4 only is there in Bihar and Uttar Pradesh and also in Gujarat, but it is not so. So both the tropical race 4 and race 1 are attacking the cavities in central and northern part of India. This we have already sent for publication plan. This is going to be published soon. This is a PCR confirmation of race 1. And this shows that you can see in Uttar Pradesh both tropical race 4 and 0125 race 1 are there. In Bihar, tropical race 4 alone, and in Gujarat 0120 belongs to race 1. And this phylogenetic analysis, when we have carried out this race 1 0125 and 0120 grouped together with other FOC race 1 isolates, and race 4 grouped separately. This shows clearly the presence of the both race 1 and tropical race 4 in Uttar Pradesh and Bihar. In other areas, only race 1 is there in Cavendish. These are all the FOC attacking the Cavendish. So in abroad, totally in the world, more than 20 different countries are having the tropical race 4. The blue indicates the presence of race 1, not in Cavendish, but in other varieties. But the red one indicates the presence of tropical race 4 in Cavendish. Recently in Colombia, this was recorded. And how to diagnose it? This is a field view, and this is the field, banana field devastated due to fissure mildew disease. And this is in Uttar Pradesh. You can see the severity of fissure mildew disease. And this yellowing, everybody may be knowing, but for the students, just I have shown the yellowing of the leaves, external symptoms. And this is the pseudostem splitting and the death of the plants. And the internal discoloration in the xylem vessels, internal symptom, and this is in the calm. And you can see the younger plants, even 15 days old plants are attacked due to fissure mildew disease. You can see the fissure mildew disease at the younger stage itself. And recently, the scientists from Australia, they have identified the presence of fissurium in the leaf regions. This may be the airborne also, sometimes the fissurium mildew. The sporadic and other things present in the petiole and leaf regions. Then we have developed the TR4 and race 1 specific molecular markers, which is specific to Indian FOC isolates. Because when we subjected the TR4 markers available abroad, this is not amplifying very correctly. So we wanted to develop ourselves the molecular markers specific to TR4 as well as race 1. So the targeting the SAX1A genes, this is common to both race 1 as well as race 4. So for race 4, tropical race 4, we have developed a molecular markers targeting this SAX1A gene, effector genes, and we have successfully developed a standardized the marker. And this is about the confirmation and using different isolates. And besides, we have developed a LAMP method for the detection of tropical race 4. And just now, Dr. Bhatt has shown all this. I need not explain this LAMP method, which are developed for FOC TR4. And we have identified the differentials also, because now that the days has gone, that race 1 should attack only the Rastali, Carprovali, and not Cavendish. But because of our findings and this system classification of race based on the differentials, what we have studied, it has gone now. So we wanted to identify the new differentials which can differentiate the race 1 from the tropical race 4. These 4 varieties, Carporochacryli, Tongat, and Atrusingan, and Monogat. This is all these are resistant to race 1 but susceptible to tropical race 4. And these the 3 differentials are resistant to tropical race 4 but susceptible to race 1. This we are validating under glasshouse condition. And host resistance, we have screened more than 300 germplasms, 260 to 300 germplasms under for race 1 as well as tropical race 4 under field condition, hotspot conditions in Theni as well as Bihar. And this is with regard to race 1, about 55 to 60 germplasms were found to be resistant, that is immune, highly resistant to race 1. And these are all the different accessions you can see here. And this one is about the diploids, which we can use it for breeding programs, and this is about the triploids, which we can recommend to the farmers. This is very important. Srimanti, Manjagaji, these are all the Cavendish group, which we can recommend to the farmers wherever the fishery milt is there, affected by race 1, which are very severe in central part of India. And this is about the race 4. Race 4 different. We have identified about 60 accessions which are resistant to tropical race 4. This is about the diploids, and this is about the triploids. You can see here Jahaji, Manjahaji, Borjahaji, which are common to both race 1 as well as TR4. So these 3 varieties are very important for the distribution to the farmers wherever the problem is there. And this is about the pictures. You can see the Manjahaji, very good bunches. And you can see the Borjahaji, very, very big bunch, and the Williams also. This is very dwarf variety, but very good variety. And in abroad, only GCTCV 218 we recommend, because this is there everywhere in Philippines, Mozambique, wherever the problem is there, and they are getting the 218 and planting it. And but this is not completely resistant. This is moderately resistant, but give very good bunches. And this is in Mozambique. They grow GCTCV 218, which are moderately resistant to banana, fishery milt tropical race 4. And this is in China. They have got ZJ9 and 4, which are resistant to tropical race 4. And the transgenic plants also available from Australia by transferring this RGA2-2 gene into the Cavendish, and it has got very good effect on this resistance. It has got very good resistance against tropical race 4. And biological control, we have worked on biological control. We identified very good.
Your time is out.
Yeah, only 2 to 3 minutes.
Yes.
So we identified some trichoderma, then some bacteria, Pseudomonas, and some botanicals also. This is GIMO. And we have bioprimed the banana plants, and we have evaluated under field conditions, and we have got a very good bunches in the abandoned field, because the farmer has abandoned that field due to fishery milt. So in that we have conducted the field trial and got very good bunch because of our application. You can see between treated and untreated, and this is about the botanical. And yeah, and for the tropical race 4 also, that is about the race 1, and this is about the race 4. And we identified a very good biocontrol agents, and we have developed a mass production methods also using cheaply available material. We're using the farm and manure. Within 5 days, you can develop you can mass produce a trichoderma, whatever the quantity you want. So this we have demonstrated in Bihar, and you can see the very good growth. And we have evaluated in under glasshouse condition. You can see here 1.5 on a 0 to 5 scale. We have got disease score due to our consortia of bioagents, and you can see here the growth promotion, the calm as well as the plant. This is in UP, and we have conducted field trial, and we have got a very good control management of this tropical race 4. And this is in Gujarat. More than this, we have applied after the infection has picked up. After 4 to 5 months, we have applied this biocontrol agents, and in that also we could get more than 57%, about 57% reduction in fishery milt incidents, particularly the race 1 in Gujarat. And this is in UP, tropical race 4. You can see very good bunches, but here the splitting and other things we observed. And apart from that, we have developed the 4 different bacterial consortia, which could control the disease completely. You can see 100% reduction in incidence under glasshouse conditions. And this is about the 4 different combination of a trichoderma of endophytic and rhizosporic nature. This has also recorded 100% reduction in disease incidence. So apart from biocontrol agents, we have to go for some intercropping with tapioca and other things. And this cover crop also can manage the disease, and growing paddy for 1 year and then go for banana cultivation. We can grow banana effectively, at least for first year. Second year it will pick up the symptoms, but first year the incidence will not be there. We have done some proteomic analysis also. In that, we identified 5 different unique protein present only in TR4, and this one is involved, particularly this one is involved in the infection, pathogen infection, and this we have identified through proteomics. And to conclude, this fishery milt, both the tropical race 4 and race 1 are spreading across India in cultivar cavities, and not alone the TR4, but the race 1 is also spreading very fast, even more than the tropical race 4. So we have to sensitize the people, all the stakeholders, and TC industries in that case, because the contaminated TC plants spread the TR4 and race 1 very fast. So the TC industry should play a major role in containing the spread of the disease. With regard to the management practices, crop rotation with the paddy, use of disease pre-planting material, and improvement of soil health, and use of effective biogens, and following good agriculture practices, these will definitely useful for the management of the disease effectively. Thank you very much for giving the opportunity. Thank you. Dr. Mayes?
Yeah.
Yes.
This
I have completed.
Chairman, sir.
topic is open for discussion. Dr. Thangavelu.
Yeah.
This race 1 and race 4 is now available. How do you differentiate symptomatically? Is it a difference is there?
No, okay. Symptomatically we cannot differentiate, but it will cause the death of the plants quickly, the tropical race 4 compared to race 1.
What do you think that race 1 is attacking now Cavendish?
Yeah.
Is it a mutation happened or a hybridation between race 4 and the race 1?
Yeah, that we have not analyzed, but the whole genome sequencing says that both are different. Tropical race 4 and race 1 is different, but yeah, this is different and this is different. Race 1. The already whatever the 0124, 125 and 1220 available, this has developed the capacity to infect the cavities because of the monoculture of banana plants in India. So wherever the bananas are grown continuously in the same field for a long time, the FOC race 1 present in the soil developed the capacity to infect the cavities. That's what the analysis says.
No, that capacity, see, virulence acquiring, that is due to the vegetative compatible strains which are hybridized. Not entire genome may be different.
We have not observed the common epitope genes present both in TR4 and race 1. So this is separate and this is separate.
So now you should highlight that one because you are the leading and scientist for this.
Yeah.
You should bring out that information that which are the genes responsible differentiating these two races.
Yeah. That's what we are working on. That already this draft genome has been published in plant disease, and we are going to write a full paper on that, comparing the TR4 as well as race 1.
Any more question? Then we may go to next presentation.
Hello, sir.
Yes.
Hello, sir.
Yeah. Hello.
Hello.
Yes. Loganan, who's?
Harish.
Yeah, Harish, ask. Ask Harish.
618.
Hello. Hello.
Talked about 618.
Yeah. Not
Can you differentiate the two races?
61. Hello.
Yes, sir.
Yeah.
Can you differentiate the one and with the TR4?
No. Both are the SIX1. both
Yes, sir.
this is present both in race 1 and TR4. We cannot differentiate, but yeah, both are there in race 1 and TR4, SAX1. The variation may be there.
He told it can design markers based on 6 famous.
Yeah, variation is there. Variation, based on the variation we have developed, variation is there. We can differentiate. Yeah. But SAX1 gene present
Thank you.
both the categories, but based on the but it has
Yes,
got
sir.
variation. Yeah.
Okay. Thank you.
Maybe next we ask Dr. Gururaj.
Yes, sir. Good afternoon.
Yeah, thank you, Thangavel, for your nice presentation and then continuing this emerging research on this TR4 and saving our banana farmers. Keep continuing on this line. Now we will invite Dr. Gururaj to speak on this assigned topic. Dr. Gururaj, please.
Yes, sir. Yes, sir. I will start. Let him close, sir.
I request all other than speaker to mute yourself so that it will not create any disturbance.
No, sir. Thangavelu, I request Dr. Thangavelu, sir, to close the meet. I am unable to share the screen. Hello. 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 Hello.
You.
Hello,
wait.
sir.
Wait. Thangavelu,
Okay.
please stop your screen sharing. You stop it.
Okay.
Dr. Thangavelu.
Yeah, I stopped it.
Yeah. Gururaj, now you can start sharing.
Yes, sir. I start. Idala. Okay. Good afternoon, sir. Chairman, sir, Dr. Krishna Reddy, sir, Makesh Kumar, and all the plant pathologists. I am going to speak on the chickpea, particularly the disease, the wilt of chickpea caused by Fusarium argyrosporum farmer species saccharis. The talk is on the molecular diagnosis, variability, and what is the prospects of plant growth promoting microorganisms for the management of this disease. So this is about the introduction. As you know, chickpea is one of the most important pulse crop cultivated all over the world, and the losses they range from 2 to 55%. And the first time it was reported by from India by Butler way back in 1918. And with respect to the particularly the molecular characterization, the variability of the pathogen, very less work has been done, and also particularly the management. The farmers mainly depend upon the cultural practices as well as very less seed treatment with chemicals, and also the PGPR. They are not at all using it. So keeping these points in view, and we thought of having why don't to study the variability of pathogen, and in that context, so what could be the variability and what are the management practices, how we can exploit PGPR for the management of this disease. And as you know, it is a age-old disease. The typical symptoms, the drooping and drying, followed by the death of plant. If you cut open the stem, particularly root, you will see the vascular discoloration. So this vascular discoloration is due to the fungus Fusarium argyrosporum saccharis, which is going to plug the xylem vessels. As a result of that, there is no movement of water and food to the above ground parts. That's why the plant gets killed. And this is the extent of damage, particularly if you go to come to North Karnataka in the farmers fields. This is a very, very severe disease until and unless some cultural practices like deep flowing and other seed treatments are not followed. So coming to the pathogen, it is a Pyromycetes fungus culture. It's heavy branching Mycelial and chlamydospores, and it also produces macro and macroconidia. And we thought of studying the diversity of this pathogen. And what we did was we have collected around 60 isolates of Fusarium oxysporum farmer species assays from different farmers field, covering nearly 12 districts of North Karnataka, and they are isolated and designated as FC1, FC2, and like this up to FC60. And when we study the colony characters with regard to the shape, margin, growth pattern, texture, then color of colony, mycelium, pigmentation, and sectoring, and we could see a lot of variation, particularly shape, irregular, somewhat regular. So many 4 to 5 shaped the isolates were noticed. So likewise, I'll summarize this table because it's a big table. And this is the photograph showing the variation with respect to culture grown on potato, dextrose, agar medium. Again, some other culture, some other isolates, and this is the one which I wanted to highlight more. You can see here in the first petri plate, the culture growth of culture was very regular. Here it is somewhat irregular, some smooth culture, then serrated margins. Here it is a circular, feathery, cottony, velvety. So all these kinds of different types of growth of colony was observed when in different isolates of the Fusarium oxysporum sesseris. Coming to sectoring, you can see the sectoring also. So different kind of sectors. So as you know, the sectoring is an indication of the variability in case of fungi. So that is very well proved, and you could see the sectors with reference to different isolates. If you summarize the results, the particularly colony growth, you can see here, I have categorized the colony growth into 5 different categories, right from very slow growing, medium growing, fast growing, and very fast growing. And you can see 51 isolates, they were very fast growing with a radial growth of 75.1 to 90 mm. And 9 isolates were fast growing, and the rest of the isolates, they did not belong to any category. And all the 60 isolates, either they are fast growing or very fast growing. The coming to pigmentation, you can see here also different kinds of pigmentation in the colony character is noticed. Right from a creamish white to white, light orange to deep orange, light yellow to deep yellow, light brown to deep brown, pinkish to red, light to deep purple. So in different isolates, 25 isolates, they belong to first category, 5 isolate second category. Likewise, maximum 23 isolates belong to light brown to deep brown category, followed by the first first category. Whereas in case of light to deep purple, of course, there were no isolates which categorized into last category. Coming to, this is the kind of pigmentation that we have noticed in the different isolates. So you can see FC1, FC4, FC5, all are different. Then coming to mycelial color is also different in different isolates. So they were categorized into whitish, half-white, light orange, and lilac. 37 isolates they were categorized under whitish. 15 isolates off-white, 6 isolates light orange, and 2 isolates lilac mycelium color. You can see the representative isolates here in this slide. The first petri plate is white colored mycelium, and it is off-white. Second petri plate, followed by somewhat light orange, and in the fourth petri plate, you could see light pink growth of the fungus in the form of mycelium, and lilac colored mycelial growth in case of last plate. So similarly, based on the mycelial characters, we could group the 60 isolates into 5 categories like fluffy, moderately fluffy, partially oppressed and scanty. And of course, 28 isolates, maximum isolates, they belong to fluffy growth. And you will see what is fluffy, what is moderately fluffy in the next slide. So this is a fluffy growth of fungus in the petri plate on potato dextrose agar medium. This is moderately fluffy and partially oppressed, and then scanty. This is the diversity with respect to cultural characters in the 60 isolates of Fusarium oxysporum sesseris, isolated from different geographical regions of North Karnataka. Then coming to the morphological characters, particularly we studied the size and subtation of macroconidia, then microconidia as well as mycelial width. Again, it is ranging, it differs with different isolates. And to summarize this, you can see here we have grouped isolates into 5 groups: very small, small, medium, large, and very large. And maximum isolates, around 22 isolates, they belong to medium sized microconidia, about 15.1 to 20, which range between 15.1 to 20 micrometers. So like this, again, shape and type of chlamydospores color also varied with respect to the isolates. And you can see 60 isolates. Finally, you can see the picture here, figures, how these conidia size and the shape of conidia varied with respect to different isolates. Again, this is a chlamydospore. Then we went to knowing this, there is a variability with respect to cultural characters and then the morphological characters. We have selected 11 isolates. Isolate number FC9, FC10, FC12, 18, 24, 27, 34, 38, 42, 46, and 55. And these isolates were selected on the basis of their distinct character. So distinct characters in the sense, the mycelium may be of different color, the macroconidia of higher size, microconidia or lesser size. Like this, we have selected distinct 11 isolates. And then we went for the PCR analysis, and these are the sequences of these 11 isolates. And this is a neighbor joining dendrogram of isolates of Fusarium oxysporum caesaris with other reference isolates. And this is a similarity coefficient of Fusarium oxysporum isolates based on SSR analysis. And you can see the polymorphism between the isolates with respect to SSR primers. And this is the dendrogram. And after that, then we went for studying how best we can use some of the native bioagents like Trichoderma, Pseudomonas, and then Bacillus for the management of disease. Because in northern Karnataka, it is only cultural practice that is deep flowing and the seed treatment. Except this, so no other practices are recommended or either recommended or followed by farmers for the management of this disease. And keeping the sustainable agriculture and then the organic agriculture in view, and we thought of testing some of the isolates, particularly the popular ones, Trichoderma, the Sclerotian Pseudomonas, and Bacillus. And what we did was we isolated 10 isolates of Trichoderma, 10 isolates of Pseudomonas fluorescens, and 10 isolates of Bacillus subtilis from healthy rhizosphere of chickpea from different geographic areas. And they were tested against the pathogen, the Fusarium oxysporum caesaris. And you can see here, all the isolates, they were able to inhibit the growth of the mycelium, the growth of the fungus. The maximum inhibition was observed in case of TR9, that is 88.51. And similarly, the Pseudomonas isolates were designated as PF1, PF2 up to PF10. Again, you can see the inhibition, particularly PF5, which inhibited the growth of the growth of fungus to the maximum extent. And so also with Bacillus, and particularly BS3, it inhibited the growth to the maximum extent. So like this is a photograph showing the inhibition of the pathogen, either by Trichoderma, Pseudomonas, and subtilis with the dual culture plate technique. You can see the maximum inhibition in case of the isolates, which inhibited maximum growth. Then we studied the cultural characteristics of these isolates of the PGPMs, Trichoderma. So you can see the mean radial growth, the colony growth also varied from white, light green, whitish green. The shape of colony also varied from circular to irregular. The type of growth fluffy and raised, and likewise the type of margin, sometimes it is wavy, smooth, and the sporulation also. And this is the cultural variability in case of 10 different isolates of Trichoderma. Then in case of Pseudomonas, they were characterized for fluorescence under UV light. All 10 isolates, they produce fluorescence and yellow green pigment. All the 10 isolates, they are rod shaped. They were gram negative, and absolutely there was no spore formation in case of all the isolates. Coming to gram reaction in case of subtilis, Bacillus subtilis, all are gram positive, rod shaped. The colored vary and texture also rough. And these are the characterization with these fluorescence. And this is the colonies of Bacillus subtilis. And with respect to different biochemical tests like KOH, catalase, casein, gelatin test, starch hydrolysis, urease, and indole test, all the fluorescent Pseudomonas isolates, they were positive, except in some isolates, they were negative for starch hydrolysis, urease test, and then indole test, and some other gelatin and casein hydrolysis. And this is how the test went on in the laboratory, the formation of string in case of KOH test, the formation of bubbles in catalase test. You can see the gram staining, the gram stained bacterial culture, casein hydrolysis, gelatin liquefaction. You can see the liquefaction of gelatin in some of the test tubes, the starch hydrolysis, urease test, and then indole test. So also the similar kind of the observations were observed in case of the Bacillus subtilis. None of the isolates, they were positive for KOH test. However, they were positive for catalase, and some isolates were positive for casein, gelatin, starch, urease, and then indole test. And coming to plant growth promoting traits, particularly the production of HCN, sidro4, indole acid, and then hydrogen sulfide, and particularly the Pseudomonas fluorescence isolates, several isolates, they were able to produce HCN, sidro4, as well as indole acid and hydrogen sulfide, and so also Bacillus subtilis. And this is presented through the photographs in the slide. Then coming to grouping of isolates on the basis of HCN production, we have grouped the isolates into strong, medium, poor, and no HCN production on the basis of the color, brick red, dark brown, light brown. And you can see three isolates. They showed very strong reaction in case of Pseudomonas fluorescens, and only one isolate, that is BS3, it showed strong reaction for HCN production. So also sidro4 production, two isolates of the Pseudomonas fluorescens, three isolates, they were strong. Only one isolate with respect to Bacillus subtilis was strong. Coming to this, the mycelial inhibition of volatile metabolites of isolates. Then we followed inverted plate technique to test the volatile metabolites produced by trichoderma on inhibition of the test fungus, that is the Fusarium axisporum ciceris. You can see TR9 again. It is able to inhibit the mycelial growth to the extent of 75.35%. So this is the photograph. And so also with respect to the Pseudomonas fluorescens.
Again, Gururaj, you have 1 minute.
Okay, sir. I'll wind up, sir. So this is the volatile metabolites. Then inhibition of Fusarium in case of Bacillus subtilis. Similar kind of results were observed with Bacillus subtilis also. Then particularly the plant growth promoting traits, particularly root length and shoot length. And what we did was we have selected very few isolates which are very effective with respect to biochemical character, the inhibition of mycelium in deer culture plate, and then we tried for different the growth promoting traits and root growth and shoot growth and seedling vigor index. Again, the similar kind of results were noticed. Finally, we went for one pot experiment. We thought of replacing the recommended practice of seed treatment as well as drenching of the chemical, particularly carbendazim plus mancozeb, and that is followed by the farmers to replace with these bioagents, particularly the treatment number, of course, the treatment number 7, which is very effective against reducing the disease resistance as well as increasing the seed weight. And this is a bio-intensive management under field conditions. Same treatment is very effective. That is seed treatment with carbendazim plus mancozeb followed by soil drenching. But to some extent, our isolates, the best isolate, they have also performed well. And finally, I could conclude that there is a variation with respect to morphological as well as cultural characters in 60 isolates, particularly the PCR amplification with ITS1 and ITS4. They are able to produce amplified product size of 550 to 600 base pairs. And the polymorphism was observed with all 4 SSR primers. And the maximum percent inhibition was noticed in TR9 of trichoderma, PF5 of Pseudomonas fluorescens, and BS3 isolates of Bacillus subtilis. And some isolates, they were able to, they are positive for KOS test, Kaplan test, and then indole test. For management, this treatment, I want to highlight the treatment, seed treatment with TR9 at the rate of 5 gram per kg, followed by Pseudomonas fluorescens 5 gram at the rate of 5 kg seed, followed by soil drenching with carbendazim plus mancozeb, that is available as Sprint at the rate of 3 gram per liter, is a little bit, is quite effective. Not effective as we see the first treatment, that is the treatment which contained the seed treatment as well as the soil application of soil application with chemical. But we can replace chemical for seed treatment with our the consortium, that is a trichoderma as well as Pseudomonas. So this is my team of students who work for this for so many years, and that's all I want to present. If you have any questions, the topic is open for discussions.
Thank you, Dr. Gururaj, for excellent presentation. The topic is open for discussion.
Okay, sir.
Gururaj, you can stop screen sharing.
Okay, sir. Okay. Shall I stop, sir?
Yeah. Because questions you can answer it.
Okay, sir.
Yes. Makesh.
Yes.
Makesh ji.
Yes.
1 or 2, then we go ahead for the next.
Yeah, that we are waiting for questions. If anyone.
Yeah. Krishna Reddy, sir.
Yeah.
Fine, sir.
Okay. Dr. Gururaj, this we have now different crops with this collected trichum. Whether this diagnostic technique you developed this will be species specific or it can work across the thing?
No, sir. As far as this is a general primer, sir, ITS primer. It can work, sir. It can. Hello, sir.
Primer is not.
Hello, sir.
ITC primer, ITS primers are not specific.
You know, sir, it is a general primer, sir. That's what I'm saying. It is a general primer, sir.
No, it can work, means it is a common, any fungus.
Yeah.
Sequencing only you can bring it down whether it is this one or not. Not
amplification
will not bring it down.
Yes, sir. Yes, sir.
Yeah. Okay, if no more question, I think I thank all the key lead speakers, 3 speakers. They have made excellent presentation. Now we will move to the oral presentation. Before that starting, I would like to know whether Swarna Lakshmi is there. Because I didn't find the name in that participant list. Joined. Lakshmi? No, okay. Let us see whether. Totally we have 5 oral presentation. And as communicated earlier, we have 6-minute presentation and 3 minutes. So please keep in time because this oral presentation it will be evaluated by the committee. So best wishes to all the oral presentation speaker. Let me first, sir, we can start that. Yeah. First, now I will invite Praveen Thomas. He will be speaking on cloning of antisense replicase gene of Sri Lankan Cassava mosaic virus. Are you available? Aip?
On the website.
Yes, yes, sir.
Yeah, you
Yes,
share
sir.
the screen and then start your presentation. You open the presentation and start scanning. Yeah. Go to full slide view. Yes. Yes, you can start your presentation.
Let me present my work cloning of
You are muted. Aip, you are muted. Huh? Aip?
sir.
Your voice is not coming.
Sir, and the title.
Okay, then we will come to next. You can change it.
Cloning.
By the time, huh? Are you able to continue?
Makesh, he has unmuted now. Let him continue.
Yeah, you continue, Aip. Illa, voice varnilla.
He is not speaking.
Sir.
Yeah.
Yes.
Yeah, speak.
Sir.
Yes, we are hearing you. You talk. You are don't mute
muted,
it.
again muted. You mute.
Move your cursor from the mute unmute, because when you press it, you get muted.
Yes. Now it's okay.
Let me.
Yeah, start.
Let me present my work cloning of
Go to full screen.
Anderson's.
Okay, continue.
Let me present my work cloning of Anderson sequence of replicase gene of Sri Lankan cassava mosaic virus. Cassava is a tropical food crop, which is the third largest source of carbohydrate. The global production of cassava is over 277 million tons. And constraints is cassava mosaic virus, cassava mosaic disease. Here is the field of the cassava mosaic disease plants. And this is a electron microscopy view of Gemini virus. There are 11 cassava mosaic virus which is affected worldwide. In India, there is Indian cassava mosaic virus and Sri Lankan cassava mosaic virus. In Kerala, Sri Lanka cassava mosaic virus is common, and in Tamil Nadu, it is ICME was also seen. Indian cassava mosaic virus and Sri Lanka cassava mosaic virus is a circular single-stranded DNA virus. It has bipartite genome, DNA A and DNA B. The genome size is 2.7 kilobase, and it is transmitted by whitefly. The viral infection leads to 25 to 88% yield reduction in India and 24% in Africa. CMD results in curling of leaves, chlorosis, leaf distortion, and turning of growth. This CMD is a serious constraint in cassava production. So the farmers will prefer the susceptible varieties. Due to whitefly, we cannot produce virus-free plants. Hence, we have to develop transgenic lines in this cassava to protect against CMD, incorporating hairpin siRNA construct delivered from pathogen. The objectives are identification and selection of cassava mosaic virus, and then to design the forward primer and reverse primer of the rep antigen strand. Next, to clone the rep antigen strand Sri Lankan cassava mosaic virus, and the transformation of E. coli with rep strand of Sri Lankan cassava mosaic virus. The rep strand was selected using integrated DNA technology software based on these criteria. And then antigen strand was selected by accepted candidate more than.
Devappa sir, please mute it. Devappa sir, please mute yourself.
And then the sequence is used to as a template to design the primer. Forward primer is modified with HIN3, and reverse primer was modified with BAMH1. And these primers are used to amplify the SLCME positive cassava plants. And here we can see the 400 base pair that we got. And simultaneously, we have isolated blue script plasmid from blue script vector. And we can see the bands over here. And then the double digestion of vector was done, and we can see double digestion using HID3 and BAMH1 in line 2 and 3. And then amplified strand was double digestion using BAMH1 and HIN3. We can see in the 5 and 6 line. Then a ligation was done using this digested product vector and this digested sequence, and then transformed into DH5 alpha E. coli cells. And then white colonies, which is recombinant, was selected, and then colony PCR was done. We got the 400 base pair. So we can confirm that there is the antigen strand in the recombinant colonies. Then to check the insert, we have double digested the recombinant white colony. So we confirm the 400 base pair antigen strand here. And this is the cloned vector, blue script vector. We can, here is the antigen strand we have cloned. To sum up, the siRNA target region of SLCME were identified, and the identified siRNA target were amplified using primers, and then antisense rep construct was cloned into blue script vector. In further work, the sense construct established, then preparation of hairpin construct with sense and antisense products. Then transformation of cassava with the development of hairpin construct for creating resistance against Sri Lankan cassava mosaic virus. That's all. Thank you.
The topic is open for 2 to 3 questions. I request Dr. Devappa, please mute yourself.
Hello?
Dr. Krishna Reddy.
Makesh.
Continue that. Because I have some problem with net here.
Continue.
Yeah. Any questions?
Okay.
Okay, then.
No
Yeah,
more.
we can move to the next speaker.
Yeah, next speaker is come.
Yeah. 1 minute. Next speaker. Who is that? I lost that connection here.
Hello?
Yeah, next one. 1 minute, 1 minute, 1 minute, 1 minute.
Hello, sir?
Yes, next Kaveri Nathan. Are you ready?
Yeah, hello?
Yeah, you can start presenting.
Sir, able to hear, sir?
Yeah, we are able to hear you.
Okay, thank you. Okay, good afternoon to all. Today my presentation is on candidate genes for the diagnosis, monitoring pathogen virulence and management of sugarcane red rot disease caused by Colatotrichum fulcatum. Sugarcane or Secarum officinarum is an important commercial crop and its cultivation is threatened by biotic and abiotic stresses. Of the biotic stresses, diseases caused by fungus, bacteria, virus, and phytoplasma play a crucial role in limiting its cultivation. Of the fungal disease, the most destructive one is red rot, which is caused by Colatotrichum fulcatum, and it can cause an yield loss of up to 100% in endemic locations. This results in the elimination of allied commercial varieties out of cultivation. Even though the varieties were released with resistance, frequent breakdown of resistance was observed due to the emergence of newer isolates of Colatotrichum fulcatum. And these are all the external and internal symptoms of red rot. So at different periods of time in our lab, we isolated different isolates of Colatotrichum fulcatum, and they were characterized by cultural and pathogenic means, and also by vegetative compatibility grouping, and also based on the production of enzymes and toxins. So at molecular level, a set of 150 Colatotrichum fulcatum isolates were categorized using 5.8 as ITS primers, and from that representatively a set of C fulcatum isolates were taken, and the multilocus analysis was performed using conserved gene sequences like actin, colmoitilidin, GPDH, and 5.8 as ITS primers, which grouped the isolates into a major group and a minor group. The major group contains most of the virulent isolate, and minor group contains the least virulent isolate. Two isolates were selected for further studies. One is a sporulating and virulent isolate, and another one is non-sporulating and least virulent isolate. the virulent isolate was taken from group 1 and least virulent isolate was taken from group 2. So, in order to identify genes responsible for pathogenicity and virulence, the two sets of isolates were subjected to proteomic and genomic approaches. In proteomic approach, all the three sets of proteome, that is the mycelial proteome, secreted proteome, and the fungal proteins expressed during host pathogen interaction, was analyzed by 2D gel electrophoresis. Unique proteins that was expressed alone in CF671, which is the virulent isolate, was subjected to multitask analysis, and from the results it was found that 27 mycelial proteins, 16 secreted proteins, and 27 fungal proteins expressed during host pathogen interaction was related to pathogenicity and virulence. Under genomic approach, suppression, subtraction, hybridization was employed among the two selected isolates, and from the results, differentially expressed transcripts was obtained. Most of the that is the major differentially expressed transcripts related to Colothotrichum graminicola, and minor was related to Fusarium fusiculae. The obtained proteins were classified, and it was classified into 14 protein families, and the peptidase was classified into 6 peptidase families, and the transporters obtained was classified into 8 transporter families. Also, it was found that of the obtained differentially expressed transcripts by SSH, 27 were related to mycelial, that was obtained in 2D gel electrophoresis, and 8 was related to secretome, and 12 was related to host pathogen interaction. Also, 24 pathogenicity related genes was identified by means of genomic approach. So, from the pathogenicity related genes obtained, the following 5 genes which were already proved as pathogenicity related genes in different fungi was selected, namely the methyltransferase, bis1 family protein, 3-isopropyl malate dehydrogenase, HSP20-like protein, and cytochrome P450 protein was taken and subjected to infanta expression studies. In this work, a set of 10 different isolates varying 10 different red rot isolates varying in degree of virulence was selected and inoculated in the standard susceptible red rot cultivar, and the samples were collected and the expression pattern was studied up to 10 days post inoculation. From the expression studies by qRT-PCR, it was found that the expression of the selected pathogenicity related genes was high or maximum at 10 days after inoculation in the virulent and least virulent isolates, whereas its expression was low or minimum level in the less and least virulent isolates. The same pattern was observed in all the 5 genes taken for the study. So, after studying the expression pattern of different genes, cytochrome P450 was taken as it is a potential target site for azole fungicides. Since nowadays azole fungicides is recommended for the effective management of red rot, so here the expression pattern of cytochrome P450 under the influence of azole fungicides was studied at in vitro and in vivo conditions. In vitro conditions, it was clear that the expression of cytochrome P450 was completely arrested by using azole fungicides like tebuconazole and propiconazole, whereas it was not observed in, or the expression pattern was observed in thiophanate methyl. The same thing was observed in the pot culture and field studies, whereas the sets were treated with fungicides in pot culture and the standing canes were drenched in field studies. Here we can able to see there was no expression of the cytochrome P450 after the sets or canes was treated with the fungicide. So, and conclusively saying, we can say that the azole fungicides specifically target the cytochrome P450. So, upon summarizing the results, the selected genes can be employed for early diagnosis of the pathogen and assessing virulence of newly emerging isolates, and the selected genes can be functionally validated at molecular level for the management of disease. Also, the identification of potential inhibitors and fungicides for the selected genes can be helpful in conventional management of the disease. Thank you. Thank you, sir. Next, sir.
The topic is open for discussion.
Hello.
Questions? Yeah.
Topic is open for discussion. 1 or 2 questions. Hello. Anybody have any clarification?
Any potential candidate for virulence?
Hello, sir.
Hello.
Sir, please say.
Any potential candidate for virulence? For virulence.
All these 5 genes are related to virulence only, sir, which were already confirmed in different fungi, and we can able to see the expression pattern, that is the virulent and least virulent isolates so maximum level of expression, whereas the less virulent isolates so minimum level of expression. That thing we can able to see in our experiments or works, sir. Hello. Sir, able to hear?
Next.
Yeah.
Yeah. No more question, then we can move to the next presentation. Kavya, you can stop
Any
sharing
other
the
questions?
screen.
Okay, thank you.
Makesh, we can go for next presentation.
Yes, thank you, Kavya. Next, we request Dr. V. Sendilvel to talk on T-shaped spore trap indices for maize Terticum leaf blight disease forecast. Dr. Sendilvel, please.
Thank you. Spore trap.
Dr. Sendilvel.
Makesh, his voice is not coming.
Sir, muted, sir. He has muted.
Cover it. Sendil, you unmute.
Oh, sir, now it is audible, sir.
Oh, yeah.
Okay, sorry. Okay. Okay. Oh, sorry, sir. Good evening to one and all. I'm going to present the new innovative approach for the management of disease. The T-shaped spore trap is indices for the maize Terticum leaf blight disease. You know that the Terticum leaf blight is caused by Exorahilum terticum. It is a foliar pathogen, airborne conidia, rapidly spread and cover the entire canopy, maize canopy, and cause the 100% yield loss under uncontrolled condition. Since the pathogen is a necrotropic and a rapid progression, pathogenesis, it totally devastates and during the maturity stage, especially the stage of the occurrence of the disease on 45th days to 65th days. In between these days, that environmental factors coincides, the sufficient inoculum is available. The rate of the spread is very severe and causes the severe loss in these maize cultivations. Then to manage the disease, that objective was designed how prevention is better than the cure, how to prevent the disease before occurrence or before spread of the disease, so that the trap spore trap is one of the device to assess the spore load in the atmospheric air. Since it is a airborne conidia, Exorhylum tursicum, if you monitor the spore trap, we can predict when the disease will occurrence while coinciding the environmental factors. So that the T-shaped spore trap was designed. Then based on the trapped conidia, the weather parameters were analyzed in these studies. Based on the analysis of the weather parameters and trapping the trapped conidia load in the atmospheric air, the decision support system was arrived for the management of tursicum leaflet disease. Here, the spore trap is a sketch is there, blueprint is there. It is a totally 8 feet iron rod. The 1 feet is the below the ground level. Here, the 4 platforms are there. On the platform, the microscopic slide will be lodged, seated. In the microscope slide is embedded with Vaseline. Here you see from the ground level, 30 centimeter above, the first trap platform, that is seating of that microscope slide blade is there. The next one is in the left hand side. That is 6 inch and 90-degree angle. Another one is mounted. Likewise, that eastern side, 2 platforms are there to trap the conidia, the western side. Otherwise, southern side and northern side. Likewise, that iron rod is mounted with a small platform with holding the microscope slide. The slide will be seated on this platform embedded with Vaseline. Then it will attack the airform conidia. So this blueprint is devised under the welding shop. The spore trap was designed. This is the spore trap we have designed and placed in this maize field. You see here, this first platform from the ground, the second, third, and fourth. In between this microscope slide, there is a groove to hold that microscope slide, which is embedded with Vaseline. So it is placed weekly once or twice. That microscope slide will be collected while after placing in the maize field. Then it will be observed in this image analyzer. So number of conidia, especially Exorhylum tursicum, Alindosporum tursicum, will be counted. Okay, the 4 microscope slide will be collected periodically, and the number of conidia trapped per microscopic slide more than 15 to 20 microscopic area. Number of conidia will be collected, then average will be taken. You see here the interesting finding observation we got observed in these studies. You see here, this is 8 to 9 and 10 septated conidia were observed in this microscopic 1, 2, 3. Here in this microscope slide, we have counted 3 Exorhylum tursicum conidia. In addition to that, in the same microscope slide, we have observed that is bipolaris maedis and alternaria and curvularia. There's so many pathogens are trapped. Here we have counted only this Alindosporum tursicum. Then accordingly, the disease incidence was also observed, calculated. Here that weather parameters like minimum temperature, maximum temperature, relative humidity, dew fall, rainfall, then wind speed, all the things, the weekly observation was made. It is conducted on the Kharif season. It is very interesting that the number of conidia, that is occurrence of the conidia, was started in this month of August, end of the August. Then September onwards, the conidial observation load was increased in the atmosphere. Parallelly, the disease occurrence was also recorded. Okay, in the September to October, it is periodically is increased the disease incidence. It is highly positive correlation with the rainfall and dew. Whenever the rainfall and dew increases, the number of conidia occurrence is noticed, the disease incidence is increased. You see here, the August 21, the first conidial population was counted. The occurrence of the disease was recorded on this September 4th. So within 2 weeks, while favoring the weather parameter, the disease occurrence was recorded. So this is interesting finding. Once the conidia is trapped in the field, we could alert the farmers to take up the prophylactic measure. You know, sir, now during the cyclone, while continuing the cloudy, rainy, and high relative humidity, the outbreak of the disease is very severe. Suppose if the spore count and based on the forecasting of the in IMD, we could give the perfect forecasting technique based on this spore trap. Here the rigorous analysis is, yes, sir.
Another 30 seconds, 1 minute you close it, okay?
Okay, sir. The spore load, this spore load is 2.6 to 4.1, default 0.11 to 7 days, temperature 22 to 23 degrees centigrade, maximum temperature 27 to 31, relative humidity 84 to 87. It's a thumb rule for the occurrence of the disease. So this is spore load is indices for the occurrence of the production of the disease. So the T-shaped spore trap is indicator for the production of the disease at the rate of 2.6 to 4.1 per microscopic field. Thank you very much, sir.
Thank you. It was new approach. Any quick question, because we are running short of time. Poster session has to start. So any one quick question?
So how many spore traps were established in a unit?
Yes, sir. Per acre, one spore trap we have placed.
And that too within the plant?
Yeah, within the canopy system area.
Within the canopy system. And what was the periodicity during which the slide was examined? How many hours?
It is not hours, it is on days based. Once in 4 days, we have taken the slide and we have observed it. But weekly observation was recorded in the disease incidents.
One question, Dr. Sendilwale.
Yes, sir.
Usually, the spore traps will be placed just above the canopy or crop.
Yes, sir.
So you are telling like inside the crop also you can put the spore trap.
Yes, sir.
So is it inside the crop or above the crop level?
So that the trap was designed up to 7 meters from the ground level. You could see the spore trap. This the above the ground canopy, 30 centimeters there, sir.
Yes.
This is a top. You see here the thank you slide, the left hand side. It is very clear. The top of the platform is there to collect the above the canopy. So the mid of the canopy, 2 more, because the incidence is mild incidence was recorded on 40th day and 45th day. Even 25th day also some initial occurrence was recorded. So at that time, the height of the crop will be 2 feet, sir. 2 feet. So that to collect the spore in the up to the height of the crop, it was designed 1 feet, 2 feet, 3 feet, 4 feet. Up to 6 feet it will be covered, sir.
That is the depends upon the height of the crop.
Height of the crop. Yes, sir.
Thank you.
Okay, thank you, Dr. Sendilwale. It was a nice presentation. Now we move on to the next speaker. You stop sharing, Sendilwale.
Thank you, sir. Thank you.
Whether Swarna Lakshmi is available?
Yes, sir.
Yeah.
Yes, sir.
You start your presentation.
Sir, am I audible, sir?
Yes, yes. Go to full screen mode.
Yes, sir. Good afternoon to one and all present here. Today, I would like to present my topic on development of loop-mediated isothermal amplification assay for the detection of bipolaris oryzae infecting paddy. Brown spot of rice is caused by bipolaris oryzae and its cilium of stage is Cochleabolus miabenus. As we all know, this disease is economically important as we know about Bengal famine, and it is of great importance. The typical symptoms of this disease includes dark brown to black spots, and it is surrounded by yellow halo. And this disease is reported to give yield loss up to 90%. The disease is mostly seen in dry and direct sown areas, rice areas. This is a microscopic conidia image, and this is a pure culture image of the pathogen bipolaris oryzae. And after confirming the pathogen, for further LAMP studies, we have chosen a highly specific and conserved gene sequence which is present only in this species bipolaris oryzae, that is glycoside hydrolase gene, family 13 protein. For further LAMP studies, we have chosen this gene. After knowing about the virulence and the potential of the pathogen, the better management practices is going for the better management practices which we have chosen is going for disease assessment and disease detection, pathogen assessment and pathogen detection. For that purpose, we have chosen LAMP assay. We know all the PCR protocols as being the standard tool for nucleic acid based assay, but for the past decades, there is a new novel technique, novel nucleotide amplification assay, which was developed by Notomi et al. In the year 2000. When compared to other PCR technique, loop mediated isothermal assay are more successful because under a single isothermal condition, under single temperature, and we are using more than 4 primers for this assay. So we can able to highly target the gene sequence. How does LAMP works? See here, after identifying the specific target sequence, we have chosen 6 primers. That is for forward inner primer, backward inner primer, forward outer primer, backward outer primer, loop forward primer, and loop reverse primer. See, once the target sequence is chosen, initially the forward inner primer goes and binds to the sequence, and then the complementary sequence is formed, and then later the backward inner primer binds to the reverse sequence, and then by using this forward inner primer and for backward outer primer, the loop structures are formed. You can able to see the loop formation here, and after by the presence of loop primer, the continuous self-hybridizing loop structures and reverse complementary sequences are formed. Because of this reason only, we can clearly see the ladder-like formation, that is concatenaric ladder-like formation pattern we can see in the agarose gel images. This schematic diagram represents the primer sequence which we have chosen for the highly specific gene glycoside hydrolase. These are the 6 genes which we 6 primer sequence which we have chosen, and after choosing the highly conserved gene, and after developing the LAMP primer through V5 primer explorer, LAMP V5 primer explorer, we have gone for several standardization procedure for this LAMP protocol. Initially, we have standardized the temperature. Different temperatures we have standardized, starting from 56 degrees Celsius to 68 degrees Celsius. We have gone through. In that, the optimal incubation temperature for the LAMP assay to identify the bipolaris oryzae pathogen was 62 degrees Celsius. In this 62 degrees Celsius only, we can able to see clear sky blue color presence in the tube, and we can able to see the ladder-like pattern in the LAMP, in the gel photo. And next, the optimization of MgSO4. The salt concentration was optimized. In that, we have chosen different concentration of MgSO4, starting from 2 micromole to 10 micromole. In that, the optimum concentration of MgSO4 salt was 6 micromole. And then we have gone for the sensitivity assay. Different folds of dilution, 10 folds of dilution we have taken, starting from 100 nanogram of DNA up to 1 femtogram of DNA we have diluted. We could able to see that presence of DNA was seen up to 100 femtogram. That is the optimum sensitivity of LAMP assay to detect the DNA concentration was seen for up to 10 femtogram. Next, specificity. We have seen the specificity of the pathogen.
Please come to the result. Conclusion, because we are running out of time.
Yes, sir. We have standardized the LAMP protocol for the bipolaris oryzae pathogen by using instead of using for conventional PCR methods, we have gone for LAMP protocol because it is rapid, simple, and it is highly specific. So we can use it for the early detection and also for several forecasting models. Thank you, sir.
Thank you. A quick question. Okay, if no question, we will move on to the next speaker, the last speaker. Yamuna, are you there? Swarnalakshmi, you can stop sharing your screen. Swarnalakshmi, you stop sharing your screen. Next speaker, Yamuna, is available?
She is available.
Yeah. Swarnalakshmi, please stop sharing your screen. Swarnalakshmi,
Yes, sir.
you stop sharing.
Yes, sir. Yes, sir.
No, still you are that thing. Close your sharing one. You go to the share screen and click stop.
Stop sharing. It is stop sharing.
Swarnalakshmi? Yeah, okay. Next, Yamuna, please start your presentation. You have 5 minutes time. Your time starts now. We are not hearing you.
Good afternoon to all.
Yeah.
I'm present. As we all know, watermelon is a major cucurbitaceous plant, belongs to the family Cucurbitaceae. It contains about more than 92% of water and is considered as a low calorific food.
Yamuna, your voice is not clear.
Sir, it's not. The yield and production of the watermelon is affected by more pest and diseases. And around the world, over 10 viruses are known to be problem in watermelon. And they are very common and cause considerable damage worldwide in severe epidemics, either in single or double infection. This picture shows watermelon plants showing chlorotic curling and stunted growth under field conditions observed in the farmers' fields in Belgaum, Raichur and Chikkalapur districts. The rowing survey was carried out in few districts of Karnataka from March 2017 to April 2018 in order to record the disease incidence of the leaf bud disease on watermelon. This table shows the percent disease incidence. The average percent disease incidence was 8.7 to 50.4%. The disease prevalence was observed in Chamarajanagar due to the continuous growing of the same crop or due to climatic conditions. These are the picture disease symptoms showing chlorotic curling and stunted growth in field condition. After inoculation of the variolous whiteflies to the healthy watermelon seedlings, the symptoms induced by watermelon leaf bud disease under artificially inoculated conditions. The molecular detection and characterization of the virus isolate. The DNA was extracted from the infected plants using CTAR method, and they are undergone PCR amplification by using the different sets of timers. Here, the gel picture shows amplification of DNA fragment of Dagamo using different overlapping primers, OY, MK, and JAMA primers. And this agar gel photograph showing amplification of DNA B fragment of Dagamo using different overlapping primers. By this PCR analysis, in all the 6 isolates collected, they shows DNA component of virus, whereas 4 isolates shows positive amplification in DNA B component, indicating these 4 isolates were bipartite Pagama infecting watermelon, and 2 isolates, Raichur and Nidabihar, they were showed no amplification of DNA B component, indicating these isolates were associated with monopartite Pagama. The infected samples undergone in PCR detection of alpha satellite using alpha satellite primers. Here, the PCR results in the successful amplification in leaf curl symptomatic adatoda leaf sample only. There was no such amplification in collected isolates. This clearly indicates the absence of alpha satellite. Coming to the characterization of the isolates, here this gel photograph showing amplification of DNA fragment by using the 3 sets of overlapping primers. This is the colony PCR showing the screening of transformants, and this picture is plasmid isolated from the transformed colonies of DNA. This clone product are undergone sequencing of viral genome, the sequences are subjected to bioinformatic analysis by using the BLAST analysis, bioedit, CLUSTAL-W, MEGAX, and STT RDP4. This table indicates the percent sequence identity of DNA component of begomavirus ranges from 93.5 to 97.4%. The sequence alignment of the tomato leaf curl Palampur virus in this study or with other tomato leaf curl Palampur viruses present in the gen bank, the sequence identity was showed that the more than 93% sequence identity was observed in each other and between the isolates, and more than 80% sequence identity observed other isolates of tomato leaf curl Palampur virus. The phylogenetic analysis of the DNA component of the watermelon leaf curl virus DNA component, this shows they form the cluster between the tomato leaf curl Palampur virus, but distinct in branch of tomato leaf curl Palampur virus. This clearly indicates that these present study viruses are the strains of tomato leaf curl Palampur virus. This table indicates percent disease sequence identity of DNA component ranges from 90.56 to 98.7% identity. The sequence alignment of the DNA component showed 95% of the sequence identity with each other and 80% sequence identity with other tomato leaf curl Palampur viruses. This phylogenetic analysis study of the present virus, they are closely clustered with the tomato leaf curl Palampur virus, and they distinct from the tomato leaf curl Palampur virus. This clearly indicates that this is the strains of tomato leaf curl Palampur virus. Coming to the conclusion, the disease incidence was ranged from 8.7 to 50.4%. Maximum incidence was observed in Chandranagar district and lowest in the Kolar district. Watermelon infected plants in field exhibit symptoms such as leaf curl, mottling, and stunted growth. Begomavirus associated with leaf curl disease of watermelon is transmitted by whitefly. The inoculated watermelon seedlings by Mycelial tabaci produced typical leaf curling, chlorosis, mottling, and stunted plant growth symptoms 12 to 15 days after inoculation. The PCR analysis revealed both monopartite and bipartite begomavirus associated with watermelon in Karnataka, absence of alpha satellite molecule in all the 6 isolates. Based on the nucleotide sequence and phylogenetic analysis revealed that begomavirus associated with watermelon in Karnataka are strains of tomato leaf curl Palampur virus. There is need to extend survey, detection, and characterization of begomavirus infecting watermelon in India. Thank you.
Over topic, any one question?
You have observed in only tomato leaf curl Palampur virus or any other viruses in your survey?
Only tomato leaf curl Palampur virus.
No other viruses you recorded in the watermelon sample.
No, sir, but
Are you-
tomato leaf bug New Delhi virus are also there, sir. But maximum is there in tomato leaf bug Palanku virus, sir.
Okay, you can stop sharing your screen. Chairman, we have to conclude the session because post session is waiting for us to close this one.
Yeah, I think the 3 lead presentations and 5 oral presentations are excellent topics covered in different techniques and crops.
No, you stop sharing your screen.
However, this I didn't find any presentation on bacteria. There should have been one bacteria or phytoplasma also, so that the all pathogens were be covering. Most of the presentations were only on fungus and virus. And the, as Dr. Bhatt has highlighted, the application of techniques should be based on the need and purpose and the cost involved and sensitivity. These points to be considered while using any diagnostic technique. But more and more diagnostics should be applied, and along with the diagnostic assay, the biological function of the host also should be seen, so that it is a holistic approach, not only by simply doing on PCR application then concluding.
ಸ್ವಲ್ಪ ಫಾಸ್ಟ್ ಆಗಿ ಮಾಡಬಹುದಾಯ್ತಲ್ಲ, ಮತ್ತೆ ಅದು ಆಲ್ಫಾ ಬೀಟಾ ಎರಡು ಪ್ರಾಜೆಕ್ಟ್
ಮಾಡಬೇಕಾಗಿತ್ತು.
Biological importance and combination with the molecular diagnostics will help in a future long way, so that we keep the face in proper identification even. And also that diagnostics not merely technique application, it should be used some purpose for, say, for screening purpose, for resistance identification, and then differentiation of isolates or strains. And the application of such diagnostics will be useful, and so that you some youngsters to know easily and pick up how to differentiate different pathogens. Such technique should be more useful. However, I congratulate all the lead speakers and oral presentation for their excellent presentation. Because of short of time, we will conclude with this. Now I thank my co-chairman, Dr. Makesh, and rapporteurs, Dr. Radhika and Harish. And Makesh, you have anything to say? And we can conclude.
No, that's all. It was all went on well, and then everybody has presented very nicely. We are running short of time because poster session is waiting, because this one has to be released. The link has to be released for them. I thank one and all for successfully participating and making this event a grand successful. Now we will meet in the poster session. Thank you very much.
Thank you, sir. Thank you, sir.
Sir, thank you.
Harish and Radhika, you.
Sir.
Harish and Radhika, you make a brief note of these points, and then we will submit to the IPS.
Okay, sir.
Okay, sir.
Kudra.
என்னோட அக்கௌண்ட் நம்பருக்கு இப்போதான் சார் எழுதி கொடுத்துருக்கோம் அக்கௌண்ட் பண்ணுறேன், அதனால இந்த 1 month மட்டும் அனுராதாவோட நம்பர் கொடுக்க சொன்னேன்.
ஹெட்டு கொடுப்பீங்களா? ஹெட்டுக்கு தான்.
ஆமாம் சார், ஹெட்டுக்கு தான்.
ஹெட்டாவது ரெக்கார்டுன்னு போடக்கூடாதுங்க, ப்ரொஃபஷனல் ஹெட்.
ஓகே.